All right, I'm going to be talking about applications of molecular biology in plant science. All the project topics you have been given has a molecular dimension. It means you can use your knowledge of molecular biology to assess your projects or to beef it up or make it better. So if you have been given a project topic under plant physiology, which involves the functions and processes in plants, for example, you have been asked to study about the effect of salts, temperature, water, stress on plant growth and development. You are comparing how different plants react to different concentrations of substances. That is under plant physiology. So what can you do? You can do what we call gene expression studies. That means you have to identify genes responsible for the various processes, like how do plants grow and what genes are responsible for their growth, what genes are responsible for them to adapt to their environment, to high salt content, to high pH levels, and to different environmental conditions. You can also do what we call molecular stress studies, in which you are trying to analyze the genes that are involved in tolerating stressors or things that can be harmful or detrimental to the growth of the plants. Then if you're under plant genetics, maybe you are under the study of hereditary genes and variations. So you can be given projects like plant breeding. You can be given projects on gene mapping, for example. So that is why you have to be involved in techniques like genome editing when you are using this CRISPR Cas9 to improve the growth of your crops or plants. Or you can do genotyping. So genotyping is determining the genetic makeup of different individual plants. You want to know because every individual is different, whether they are from the same parent or not. So focusing on the genetic makeup of each plant is what we call genotyping, but that is under plant genetics. Then if you're into plant ecology, where you have to study the relationships between plants and their living and non-living environments, you are considering plants in different communities. This is where your supervisor will tell you to go to a community to go and gather information about a particular plant, how they are diverse. Let's say the species of mango plants in Ekpoma versus the ones in Uromi. When you are giving topics like that, so how can you involve molecular biology? There's what we call DNA barcoding. So with that DNA barcoding, you'll be able to identify the different species of plants without using their morphological characteristics. You just know that, okay, through DNA barcoding, this is this particular species of this particular plant or something. So you can also do what we call epigenetics, where you have to study how the environment affects the different characters of the plant and whether these characters can be passed down to the next generation or not. That is under plant ecology. Then if you are into plant pathology, which involves studying plant diseases, their causes, their management and their symptoms, then you'll be given topics like fungicides. You'll be able to culture fungi in a lab, then the activity of some substances on the fungi. Then you also be given things like early diagnosis. That means if you can know the type of pathogen, whether it's a virus, whether it's a bacteria, or whether it's a virus that is affecting the plant on time, so that you can proffer solution to it. So you have to use things like PCRO to determine the particular pathogen for early diagnosis. You have to use devices like ELISA and some other things. Then you also have to study the host pathogen interaction. That is for that under plant pathology. Then for plant biotechnology, you have to you'll be given topics like you have to study transgenic plants. That means GM plants, genetically modified organisms or genetically modified plants, in which case you have to do a lot of studies on, okay, how you can transfer genes from one source to another across different organisms. So there's no genetic barrier or biological barrier. You can transfer genes from plants to animals and vice versa. And this is under plant biotechnology. Or there's what we also call the synthetic biology, where you have to engineer plants to produce biofuels, pharmaceuticals for industries, different enzymes. You can create hormones and enzymes like insulin and other bioactive compounds that will be under plant biotechnology. Then for ethnobotany is the relationship between plants and humans. This is when your supervisor will tell you to go to a particular community. The community, you'll be asking questions and giving them questionnaires to answer how they have been using these plants, what they have been doing with the plants, and all of that over the years, whether the plant is medicinal or not. So this falls under ethnobotany. So how can you employ molecular biological techniques under this area in plant science? So to do that, you have to do what we call molecular phytochemistry, where you have to identify the active compounds in those plants you are trying to assess by using what we call gene expression studies. Then you can also go into conservation genetics. And the conservation genetics use a lot of molecular techniques involved in genetic conservation, includes DNA sequencing and phylogenetic analysis, where you have to look at the evolutionary relationship between organisms. This one, you are not just depending on what the people are telling you in the community. You are using their genome. You're analyzing their genome to see how they are connected from time past. So that is what we call phylogenetic analysis. And so we also have genetic marker analysis and whole genome analysis, when you analyze the entire genome of the organism. So that is under ethnobotany. Those are the molecular techniques you can use for ethnobotany if you are given a project under that. And so basically, when you are giving any plant biology work, you have to first of all extract the DNA from the plant. Then if you can sequence it to know to identify the particular species of that plant before you can carry out other activities based on your peculiar research topic. So we also have for horticulture. For horticulture, you can use what we call marker selection to identify the proper traits or the traits that are useful in tissue culture experiments. Like when you start tissue culture, even from that callus formation from the plantlets, you can identify the particular trait that you want. For example, if you want sweet fruits, you can know that, oh, this particular plantlet is going to produce sweet fruits, even without allowing the plant to mature. So all these molecular marker selection is another technique you can use for tissue culture. That is for under horticulture. And then of course genetic enhancement, crop improvement. You want to improve the color, the yield, the shelf life, and various other needed traits. You can do this transgenesis, where you can transfer desirable genes across plant species. Like I said, it has no biology barrier. Then for forestry, you can do sequencing analysis, you can do molecular breeding until you get your enhanced breed, depending on your requirements or what you need at that moment. For agronomy, for agriculture, crop improvement, and then you want to also know the particular microbe that is responsible for proper growth of the plant. So let's say there is a particular microbe that can fix nitrogen from the atmosphere, and you know that nitrogen is important for the plant. So you can genetically modify that microbe, that nitrogen fixing bacteria, so that you can get the best or highest amount of nitrogen from the atmosphere. It's basically your crop improvement under agronomy. Then of course for plant molecular biology, We have what we call transcriptomics, where you have to take all the profile of the RNA. We have different types of RNA. You take their profile and then you assess them. Then for proteomics, just like the name signify or implies, it's all you are just doing protein analysis, because proteins are very important. Your enzymes are of course proteins, so you want to know all the proteins that are involved in a particular plant and their functions, and the ones that you do not like. That means there are some proteins that can cause damages. So this is where scientists can identify those proteins and inhibit them, thus preventing them from causing damages to the plant. For example, if a microbe attacks a particular plant, the microbe will infuse its own deadly protein into the plant. So if those proteins are inhibited, it means that they cannot affect the plant. So this is just like an overview of the different molecular biology applications in various areas of plant science. So what I want you to do is that think about your own research topic that is peculiar to you and find out how you can apply molecular biology in your research topic.